This video is going to provide you with an animated overview of what's going to happen during translation. Translation is the process of making proteins. So another way of saying that is protein synthesis. There are a few key players that we're going to discuss in later videos, but this process can be a little bit hard to imagine. And so I want this video to just kind of illustrate what's happened so far and then what we're going to talk about in greater detail as we dive into translation. So first, let's go ahead and remind ourselves how we've gotten to this point. So first, there's a molecule on the screen. Do you think that's DNA, RNA, or protein? Well, it's got two strands and there are thymines in there. We know that's DNA. So during gene expression, remember, we're going to start with DNA and we're going to make RNA, and that process is called transcription. And since RNA is only a single strand, we only need to use one of those DNA strands as a template. So remember, RNA is always going to be made 5' to 3'. So that means that this top strand here, in this example, it's the top, but it's always the 3' to 5' DNA strand, and that's going to be used as the template. Okay, so you'll notice that the RNA that's made after transcription is anti-parallel to that template strand, and it's also complementary. Notice, because it's RNA, you see uracils in the molecule rather than thymine. So if you look at that top template DNA strand, it starts with an adenine. Well, adenine always pairs with uracil in RNA. Go up against the DNA. Second base is guanine, so it better be complementary in our RNA. So we have cytosine. And you could do that the whole way down. Just remember, because RNA does not contain thymine, you're always going to have adenine and uracil base pairing together when we're talking about RNA. Okay, so once we have this RNA transcript, also called messenger RNA, or mRNA for short, the next step in gene expression is to use this messenger RNA to create a protein. And that process is called translation. And so essentially what's going to happen is the cell is going to read the messenger RNA and use that to build or to connect amino acids together. So remember, proteins are made up of amino acids, okay? And so essentially what's going to happen is, based on the instructions in the RNA molecule, specific amino acids will be connected to one another in a certain sequence, okay? And so we actually call this chain of amino acids a polypeptide chain, because technically we shouldn't use the term protein until those amino acids have all been attached together and they're able to fold into their 3D shape. A protein is a fully functional molecule. Okay, so that means it's completely folded, so it can be completely functional. So technically, this chain of amino acids is called a polypeptide chain. Okay, so you might be wondering, well, because RNA is made up of nucleotides and proteins are made up of amino acids, like, how do we read the RNA to figure out how to build this polypeptide chain? Well, let me show you. So, this is a different RNA sequence, but notice it is a single strand. It runs 5' to 3', and you see uracils in there. Okay, that's how you know you're looking at RNA. So in order to figure out the polypeptide sequence, the first thing we have to do is separate out these, group these nucleotides into sets of 3. So AUG is my first set of 3. My next set of 3 is AAU. And then we continue on until we're at the end of the piece of RNA. We have a term for these groups of 3, and that is codon. So AUG is going to be the first codon, and it's also called the start codon. Right? Every messenger RNA, at least that we'll talk about in this class, will always start with AUG. Okay, so that's why it's called the start codon. So if you're ever doing a practice problem on your own and AUG is not your first codon, then there's been a mistake somewhere. Okay, so we're going to go ahead and number these codons. So AAU will be codon number 2, and then we have 3 through 10. So this piece of RNA has 10 codons. All right, well now what do we do with all of these codons? Well, I've posted online, and what you will have provided to you during quizzes or exams is what's called a codon wheel. So let me move that onto the screen. And so what's going to happen? Oh, where did it go? Hold on a second. Sorry. There it is. So what we're going to do is we're going to look at the first codon, AUG, and we're going to use this wheel to figure out what amino acid AUG codes for. So you'll notice that in like the middle, the bulk of this wheel are A's, U's, G's, and C's. These are RNA nucleotides. But on this outer edge are the amino acids. There are 20 different amino acids, and they're often abbreviated with a 3-letter code or a 1-letter code. We will always use the 3-letter code in this class, okay? So in order to figure out which codon codes for which amino acid, you have to start in the center. So for our start codon, the first nucleotide is A, so we're going to start here at the A. Then we work our way outwards. So next is U, so we're going to work out to that U. And our third nucleotide is G, so we're going to move on out. And see how this gives me MET, M-E-T? Okay, so that's going to be our first amino acid. So now we're going to do the same process with our second codon. So again, for this one, the first nucleotide is A. So again, you always start in the middle. So there's our A. Second nucleotide is A. And then work our way out for the third one, which is uracil. Okay, and so that gives us the 3-letter code ASN. All right, I'm going to do one more with you, and then I'm going to have you do the rest on your own. So our third codon is CCG. So again, always start in the middle. So here's our C, and then G. So notice that gives us our 3-letter code of pro. And there it is. So go ahead, pause this video, and use a codon wheel to determine the amino acids for the rest of them. Now I'm going to go ahead. I'm not going to sit here for 10 minutes. I'm going to go ahead and tell you what they are. So next is going to be TYR. It's for the amino acid tyrosine, which you don't have to know their actual names. The next one is glycine. Then we have serine, proline. So notice it's okay to have the same amino acid show up multiple times in a polypeptide chain. A lot of times, a protein will be made up of a minimum of 500 amino acids, and there's actually only 20 amino acids that exist. So there is going to be a lot of repetition of amino acids in a given protein. All right, so then our next amino acid is leucine, and then threonine. So now you might have gotten to this last one, which is going to be our stop codon, because it's at the end. We need to stop translation. And you might go, wait a minute, what do I do here? So let me bring the wheel back on. Let me scooch it over. So we're doing UAA. So we're going to start here, U, work our way out to A, work our way out to the other A. And you see this circle. All right, well, at the bottom here, it says that means stop. Okay, again, that means we're done, no more. But there are no amino acids called stop. Okay, look at all these other names, y'all. Asparagine, glutamine, phenylalanine, right? Why are they suddenly going to give us an easy one like stop? Okay, so the stop codon just means this process is coming to an end. So you do not write stop for codon 10. Okay, you just leave it there. This is the amino acid sequence, okay, or the polypeptide sequence. Okay, so this is what we're going to be talking about in more depth in future videos, but I wanted you to understand how we actually read the RNA in order to figure out what amino acids we need to eventually create the protein. All right, y'all, that's it for this video. Let me know if you have any other questions.